Good evening, everybody. Today, I'll be presenting a paper titled Variation of Endoreduplication and its Potential Contribution to Endosperm Development in Rice, Oryza sativa lat. So, this was published in the journal Plant Production Science in 2019 by Hidekazu Kobayashi. So, this is a general outline of the paper published. We'll be dealing with the abstract of the paper. So, the term endoreduplication. So, it is a phenomenon by which the cells increases their ploidy. In this case, the chromatids are duplicated exponentially while the chromosome number remain unchanged. This process is initiated during the transition from the mitotic cell cycle to the modified cell cycle, which is known as the endocycle. So this endocycle, in this process, the DNA replication occurs where there is no subsequent chromosome separation and cytokinesis. That is, no chromosome separation and the cell division takes place. So this endoreduplication, this phenomena was observed in the rice cultivars, and this phenomenon was observed by two techniques, that is flow cytometry and fluorescent microscopy. So under flow cytometry, the phenomenon was detected under three parameters, and here we have taken 10 cultivars of the Oryza sativa. So the three parameters which were detected were the mean ploidy of all nuclei, the proportion of the nuclei, and the mean ploidy of nuclei. Again, moving on to the second technique, that is the fluorescent microscopy. In this technique, the things which were revealed were the ploidy of nuclei was higher in the case of intermediate region when compared to that of periphery or the central region of the endosperm. And cells with the larger ploidy were larger in developing to the endosperm. Also, the mean ploidy in developing endosperm is proportional to the cell size. Now, these are the key points which were discussed in the paper. That is, endoreduplication in a rice endosperm cell is being regulated by two phytohormones, which is ethylene and then it is the cytokinin. So ethylene, 1-methyl cyclopropene, that is an ethylene inhibitor, and 6-benzyl aminopurine is a cytokine. So these are the two phytohormones which has its effect on the process of endoreduplication. Now, the cell cycle regulatory genes, namely that is KRP1 and CCS52A. So these two are two important genes which is very important for the transition from the mitotic cell cycle to the endocycle. So if there is any change in the expression of these two genes, that is any mutation taking place in these two genes, this hinders the endosperm development. Now, what is the relationship among the three parameters which we have discussed? So you can see the relationship is that mean ploidy is equal to 3 into 1 minus the proportion of nuclei divided by 100 plus mean ploidy of nuclei into proportion of nuclei divided by 100. So this is a formula to identify or to calculate the mean ploidy. So what is mean ploidy? What does this parameter say? So this is an index of endoreduplication. And the proportion of the nuclei, it is used to estimate the initiation of the process of endocycle. And the third one, which is the mean ploidy of nuclei, is used to estimate the reiteration of endocycle. Now, what is this reiteration of endocycle? That is the repeated process of endocycle taking place in endosperm. Now, the exact role, what exactly this endoreduplication is doing in this particular cell, that remains unknown. Okay, but still, the scientists believed that it contributed to cell enlargement, increased gene expression levels, accelerated growth, and increased DNA storage. So these all inference could be formed by the experiments carried out in this particular plant. Now, what are the objectives? So the main objective of the study was to clarify the features and variations of endoreduplication in rice endosperm. And for this objective, the scientists have taken 5 Japonica and 5 Indica cultivars, and to list them, the Japanica cultivars were Akita Kamochi, Kinu Hikari, Koshi Hikari, Nippon Bare, and Sasanishiki. And the Indica cultivars were Habataki, Hokuruku 193, IR64, Kasalat, and Takanari. So these were the 10 different uh, cultivars of rice taken for the study. In 2014 and 2015, the rice cultivars were grown under natural condition in the paddy field at the Western Region Agriculture Research Center in Japan. Okay, now the relationship between the endoreduplication and the cell size was investigated to clarify whether this process contribute to the control of cell size in endosperm. Now the materials and methods. So Kinu Hikari, which was an Japonica cultivar. So these plants were grown under natural conditions. So we are taking one example in this case. So this plant were grown under natural condition in a paddy field. So say it was grown in Western Region Agriculture Research Center, National Agriculture and Food Research Organization in Japan. And after this were grown, the caryopsis were germinated in a nursery bed, and the seedlings were then transplanted into a paddy field at a high spacing. Okay, so at a hill spacing that was around 15 to 30 centimeter with 2 seedlings per hill. So that is how the caryopsis were germinated in the nursery bed. After that, chemical fertilizers were applied to the basal dressing as a basal dressing 20 days before the heading or the head formation. After that, the pollination dates of the caryopsis were recorded. Now, for the flow cytometry, 2 caryopsis per plant were obtained using the forceps, and it was immediately placed under the microcentrifugation tube and was stored in minus 80 degree Celsius condition. So for the next one, that is a fluorescence microscopy, the caryopsis at 5 and 9 days after the pollinations were fixed in the farmer solution. Now, to examine the process of endoreduplication in each cultivar, that is 10 cultivars, at a similar developmental stage, the caryopsis harvested where when the dry weight was about 25% during its maturity period, and this was used for the subsequent experiment. So as you can see in the slide, you can see a flow cytometry in the upper region, and the below here you can see a fluorescent microscopy. So these two techniques were used to identify the process of endoreduplication in the rice cultivars. Now, so statistical analysis. So the statistical analysis was carried out using the JMP11 software, and the data which were obtained were analyzed by 1 or 2 way analysis of various variants, that is ANOVA, and then measured. The mean ploidy was measured using the Tukey-Kramer multiple comparison test. So what were the results obtained in this experiment? So when the cultivars were passed through the flow cytometric analysis, it detected 4 to 5 peaks of fluorescent intensity. Okay, intensity of the nuclei which was developing in the caryopsis. As you can see in the picture which is given on the right side, A, B, and C. In this figures, you can see around 4 to 5 peaks of fluorescent intensity. So this is was seen in the developing caryopsis. Now the third figure, you can see only one single peak that is detected in a flag leaf. Okay, that was a difference which was identified when observed under the flow cytometry. Now, to clarify the spatial progression of endoreduplication in rice endosperm, median transverse section of the caryopsis were stained with DAP1 and it was later examined under the fluorescent microscope. Now, what we observed was that the mean ploidy differed among all the cultivars, all the 10 cultivars. And the lowest one of the lowest ploidy, mean ploidy, was observed under the Akita Komachi, which was a Japonica cultivar, and the highest mean ploidy was observed under Takanari, which was an Indian cultivar. So the endosperm of cultivars with a higher mean ploidy or E6P, that is the mean ploidy of nuclei, as indicated by flow cytometry, contained that more than 24 nuclei. Okay, so the cells which is seen in the intermediate region of the endosperm were larger than those of the central and the peripheral region in all the examined cultivars. So this was also uniform. So that is what we see in this figure. So the first set of figure 5 picture that is seen under the Japonica rice cultivars, and the second set is that of the Indica rice cultivars. As you can see this fluorescent images, the cells in the central region, not in the central, sorry, the cells in the intermediate region, the region between the periphery and the central region, the intermediate region cells are larger in size when compared to on the either side of the endosperm. So that was uniform in all the 10 cultivars. Now, this is the final result which we obtained in this, which was obtained in this paper. That is, as you can see, there is a mean ploidy, the proportion of ploidy, and the mean ploidy of nuclei. As in this three graph, when compared, you can see that the endoreduplication in developing endosperm among the 10 rice cultivars. So the mean ploidy of all nuclei in developing endosperm has been depicted in this bar diagram, and the highest one is seen in which we said the Takanari. This is the last bar, TA. TA represents the Takanari, and the lowest one is depicted by the AK, that is Akita Komachi. So these two cultivars were having the higher and the lower ploidy level. Now, there were few drawbacks in this study. This one was that there was a lack in the ploidy map. So this ploidy map revealed the distribution of rice endosperm cells in different ploidy classes. So that was lacking in the case of rice. But in other plants like maize and sorghum, the ploidy maps were more prominent. Even though the process of endoreduplication appeared to play an important role in determining rice endosperm cell size, there was no significant correlation between the mean ploidy in the developing endosperm and the weight of the mature caryopsis. So there was no correlation between these both factors. Now, as we said, there was an enlargement in endosperm cells. Ploidy level also increased. But this enlargement of endosperm cells associated with endoreduplication may affect the quality of rice as food, because rice is one of the major staple food which has been taken by us. So why does that quality has been affected is because the endosperm cell wall got affected. Okay, so this considered to affect the texture when the rice is being cooked. So the endoreduplication progression in endosperm cells varied among the 10 examined rice cultivars with the highest and the lowest, as we discovered, was in, as we have discussed, was in TA and AK respectively. And it was detected, this process was detected as early as 5 days after pollination in a rice endosperm, although the mean ploidy of nucleus was, when we have taken the ploidy, the mean ploidy was low during the 5 days after pollination. So the mean ploidy of endosperm cells started to increase till the date of 8 days after pollination, and after 8 days of pollination, it remained constant. So in rice endosperm, the mitotic cell division takes place, or it begins after the 4 days, um, at 4 days after pollination, and then it ceases, or it becomes stationary at 8 or 9 days after pollination. So this observation indicates that endoreduplication overlaps with the temporally with the mitosis process which is taking place in the rice endosperm. Now, the switch from the mitotic cell cycle to the endocycle starts with cells in the central region and spreads outward. So you can see the cells in the central region in the rice endosperm are also formed earlier than those in the outer region. Okay, so therefore, the cells in the central region of the rice endosperm should cease the mitosis earlier when compared to that in the intermediate or the peripheral region. As a result, the cells in the central region of rice endosperm reportedly commences the process of programmed cell death around the 10 days after pollination. Now, the nuclei in the central region retained a low ploidy throughout the rice endosperm development. Therefore, the genetic variation mediating the endoreduplication of endosperm cells may be applicable for improving the rice yield and or quality via the changes in the cell size. So these were the points of discussion in this uh, presentation. So here my presentation ends. Thank you.